Now, why intermediate filaments? Intermediate filaments are dimers between 8 and 11 nanometer. Since this is intermediate to both actin as well as the recently studied tubulin, this is called as intermediate because the values are intermediary range, which is intermediate because the diameter of the two other principal elements of the cytoskeleton is there. Actin filaments about 7 nanometer and microtubules about 25 nanometer. So, between 7 and 25 you have 11, so it is called as the intermediate filaments that have diameter between 8 to 11 nanometer. In contrast to actin filaments and microtubules, the intermediate filaments are not directly involved in cell movements. To date, intermediate filaments have only been identified in animal cells. Intermediate filaments are strong, flexible, rope-like fibers that provide mechanical strength to cut to cells that are subjected to physical stress, including neurons, the muscle cells, and the epithelial cells that line the body cavities. Unlike microfilaments and microtubules, intermediate filaments are chemically heterogeneous group of structures that in humans are encoded by approximately 70 different genes. This I have taken from FABS Letters, a prestigious journal, and this journal gives a descriptive account of intermediate filaments. You can always give a general reading if you are interested to study further. Accordingly, intermediate filaments are everywhere, right from lens where you have facalin 6 and filcellin. You have epidermal keratins, keratin 1, 2, 5, 6, 9, 10, 14 and 17. You have simple epithelial keratin in the lungs such as keratin 7, 8 and 18, 20. We also have on hair shaft and nail keratin 31 to 38 and 81 to 86. Nervous system has neurofilaments, the alpha internexin, nestin, perforin, GFFE, glial fibrillary acidic protein. The muscles contain cardiac, skeletal and smooth muscles contain desmin, syncolin and cinnamon. The blood vessel and other mesenchymal tissues contain vimentin, and finally the nucleus has laminin A and laminin B1 and B2. So, these are the different types of intermediate filaments. Now, we study what is the structure and assembly of intermediate filaments. You have an N-terminus and a C-terminus domain. It is a central rod domain containing of alpha helix with 310 to 350 amino acids. The intermediate filament proteins contain central alpha helical rod domains and N-terminal head and a C-terminal tail domains, already I highlighted. The central rod domains of the two polypeptides wind around each other, see coils or winds around each other in a coil structure to form dimers. There you have it coils and ultimately after coiling it forms dimers. Later the dimers will associated in a staggered antiparallel fashion to form tetramers. Tetramers is 2 plus 2 tetramers, and tetramers associate end to end to form protofilaments, and laterally it joins to form filaments. That's tetramer joins to form protofilament, and protofilament twines around to form filament. Each filament contains approximately 8 protofilaments. Each filament contains 8 protofilaments wound around each other like a rope like structure. It's like a coir or a rope like structure that is formed. When you talk about the intracellular organization of keratin filaments, we have a GFP photograph, a micrograph of epithelial cells stained with fluorescent antibodies to keratin, which looks brilliant green. The keratin filaments extend from a ring surrounding the nucleus to the plasma membrane. Last but not the least, we shall study a bit about the attachment of intermediate filaments to desmosomes and hemidesmosomes. First, we will go with desmosomes. This is the schematic representation of desmosomes, where the desmosomal cathedrins, that is desmoglein and desmocholine, I will highlight the desmoglein and the desmocholine, that's desmoglein and desmocholine. So, the desmosal cathedrins such as desmoglein and desmocholine link the adjoining cells to the intermediate filaments. How it links? Through placoglobin, the placofilin and desmoplakin. Now we have the schematic representation of hemidesmosome. In hemidesmosome you have integrin alpha 6 beta 4, integrin alpha 6, integrin beta 4, and they will link the extracellular matrix to the intermediate filaments through plectin. BP180 and BP230, BP180 and BP230, BP230 and BP180 regulate hemidesmosome assembly and stability, and they are used for intracellular communication purpose. Now, what are the applications of intermediate filaments? Nestin is a stem cell marker. GFAP, glial fibrillary acidic protein, this can be used as an astrocyte marker in analysis of neuronal tissues. Vimentin, very important, is an antibody that can be used as a neural stem cell marker. Vimentin is highly expressed in fibroblast, and some expression is also found in T and B lymphocytes. The expression, they are expressed in many hormone independent mammary carcinoma cell lines. Vimentin maintains shape of the nucleus, and also it maintains the organelle positions, do not dangle and move from one end to another. So, those were the applications of intermediary filaments. One last MCQ from this portion: which of the following is the most extensible filament? The four options are intermediate filaments, cytochrome, microfilaments and microtubules. Your answer obviously would be intermediate filaments. Finally, let me acknowledge the person, all those who have made this possible. Ms. Megha Dave, my student and current faculty, for preparing and compiling the CSIR net-based question banks. Dr. Darshi Bakshi, Associate Professor, School of Sciences, Navratna University, along with Dr. Elizabeth Robin, the co-coordinator for hosting this program under the aegis of Gujarat State Biotechnology Mission. Ms. Ansuya Badalkar, Joint Director HRD GSBTM, for providing this platform, for her constant motivation and support in uplifting the biotech human resource across the state of Gujarat. And finally, with due acknowledgement to my guru, from whom I learned the tenets of life sciences, Professor A.V. Ramchandran, I would like to thank all the participants and august audience for your patience. Thank you very much.